Before taking a closer look at each individual thermal method of controlling microorganisms, it is useful to first look at how temperature tolerance in microorganisms is quantified. The most widely used metric to describe temperature tolerance in microorganisms is the so-called decimal reduction time, or d-value. The d-value is always given for a particular temperature, shown here as subscript capital T. The decimal reduction time at a given temperature is defined as the time required to reduce the number of viable cells of a specific microorganism by one order of magnitude, i.e. a factor of 10, which is the same as a 90% reduction in viable cells. This means that exposing a specific microorganism to the same temperature as indicated by the subscript of the d-value for a period of time that equals the decimal reduction time, the number of viable cells will decrease by a factor of 10, i.e. a 90% reduction. If the exposure time is twice that of the decimal reduction time, the number of viable cells will decrease by a factor of 100, i.e. a 99% reduction. If the exposure time is 3 times that of the decimal reduction time, the number of viable cells will decrease by a factor of 1000, i.e. a 99.9% reduction, and so on. How is the decimal reduction time determined for a specific microorganism? This can be done quite easily by exposing the microorganism to the temperature in question and then measuring how cell viability decreases over time. Imagine that we have a population of 1 million viable cells of the microorganism we are interested in. We expose this population to a set temperature for 6 seconds and observe that the number of viable cells has dropped to 1000. Note that the y-axis has a logarithmic scale. Using this formula, we can calculate the decimal reduction time for this particular temperature. We first enter the values for our two data points. Note that I'm expressing the number of viable cells in base 10 for clarity. By taking the base 10 logarithm of the viable cell numbers, we now get these values. We then simply do the subtractions to get the numerator and denominator. After dividing the numerator by the denominator, we get a decimal reduction time of 2 seconds. We can further illustrate this by plotting a line through the two data points, again noting that the y-axis has a logarithmic scale. Now we can clearly see that a 2-second exposure time corresponds to a 10-fold reduction in viable cells from 1 million to 100,000. Another 2 seconds of exposure corresponds to yet one more 10-fold reduction in the number of viable cells, this time from 100,000 to 10,000. Yet one more 2-second exposure corresponds to another 10-fold reduction in the number of viable cells from 10,000 to 1,000.